Welcome back to this next video in which we are discussing the transportation of oxygen and carbon dioxide within the bloodstream. Okay, so we are currently discussing oxygen, and we're discussing oxygen in the venous blood. And we've seen that oxygen is going to be dissolved within the venous blood, and we've seen the concentration of oxygen dissolved in the venous blood normally. And what we've moved on to discussing is the fact that oxygen is also going to be bound to hemoglobin proteins within red blood cells. And in fact, the oxygen that's dissolved in the blood, so dissolved in the water of the blood plasma and the cytoplasm of the red blood cells, that's going to be in equilibrium with the oxygen that's bound to the hemoglobin proteins. So, we have now discussed the structure of hemoglobin. We've seen that hemoglobin is made of a tetramer of globin polypeptides, so 2 alpha globins with 2 beta globins, and to turn that globin tetramer into a full hemoglobin protein, you have to put onto each of the globin monomers a prosthetic heme B group. But from now on, I'll probably just refer to that as a heme group rather than specifying that it's specifically heme B, but it is heme B specifically. Okay, so each of the globin monomers has a prosthetic heme group, and that's now the structure of hemoglobin. So once you've got this globin tetramer with the 4 prosthetic heme groups, that is hemoglobin. Right, so what we now want to discuss is how is hemoglobin going to bind to oxygen molecules? And the way in which this is going to occur is the oxygen molecules are going to bind to the prosthetic heme groups. So the prosthetic heme groups are absolutely essential in hemoglobin. And the way that the oxygen molecules are going to bind to these prosthetic heme groups is via the ferrous cation. So when we looked at the structure, when we looked at the way in which the prosthetic heme groups attach to the globin polypeptides, I showed you the fact that they attach via these histidine residues. And we have seen that the ferrous cation at the centre of the prosthetic heme group is going to have used up 5, therefore, of its 6 coordinate bonds, its 6 possible coordinate spaces where it can interact with an atom. So it's used up 4 in the porphyrin ring, and I will just go back up to this picture. So it's used up 4 in the porphyrin ring, and it's used up 1 to attach to the histidine residue, so it has one final one to interact with an oxygen molecule, and this is indeed how the prosthetic heme groups interact with an oxygen molecule. So, let me now show you then how an oxygen molecule can bind to a prosthetic heme group of a hemoglobin protein. So, here is a oxygen molecule, and remember it will consist of 2 oxygen atoms double bound to one another. And the important thing to remember is that each of the oxygen atoms in an oxygen molecule will have 2 lone pairs of electrons. Now, of course, these lone pairs of electrons, we've heard this story before, they are centers of negative charge, and therefore they will be attracted electrically to the ferrous cation, which has a +2 positive charge. So you'll form a nice little interaction here, electrostatic interaction, and this is then how oxygen molecules can bind to the prosthetic heme groups. And now, of course, that ferrous cation has 6 of its possible interactions used up, all 6 of them, so it can't interact anymore. Right, so that means overall that a single hemoglobin protein can bind to 4 oxygen molecules because it has 4 prosthetic heme groups and each one of them can bind an oxygen molecule. Now, it's not the case that if a hemoglobin protein binds oxygen, it's necessarily going to have all 4 of the slots filled at any one time. It might have 2 or 3 or just 1 of the slots filled, so you can have those intermediate fullness, if you like. However, an important concept with regards to oxygen binding to hemoglobin is the concept of cooperativity. Okay, so we say that hemoglobin binds oxygen in a cooperative manner. Now, what does this actually mean? This refers to the fact that once one oxygen has bound to a hemoglobin protein, it then becomes easier for more oxygens to bind to that same hemoglobin protein. So let me run you through this nice and slowly. So let's say we have created a hemoglobin protein here. So we have our 4 globins and we have 4 prosthetic heme groups, and at the moment it's got no oxygen bound to it. Let's say we bring along an oxygen molecule, so here comes an oxygen molecule, and this oxygen molecule could now bind to one of these heme groups. Now, it's not instantly going to happen. There is a possibility that the oxygen won't bind to the heme groups, and the probability that the oxygen atom will bind is a concept that we call the affinity of the heme group for binding oxygen. So there is a certain likelihood that it will bind. The higher that is, the more affinity the hemoglobin has for the oxygen. The lower that is, the lower the affinity of the hemoglobin for oxygen. So let's say one oxygen does bind to, let's say, this heme group here. What now happens is that triggers a very slight conformational change in the hemoglobin protein, and the affinities of the remaining prosthetic heme groups for binding oxygen changes, and it changes so that it's higher, okay? So that if another oxygen molecule comes along, so a second oxygen molecule comes along, it's more likely that that oxygen molecule will actually end up binding to one of these prosthetic heme groups because they have a higher affinity for binding oxygen now. Okay, and that continues on. So once you've got two oxygens bound to prosthetic heme groups, there is a further conformational change, and the affinity of the two remaining prosthetic heme groups grows even more. So binding of oxygen promotes more oxygen to be uptake. So once you've got one oxygen bound, it then becomes more likely that you'll progress to having all four of the oxygen binding sites filled in that hemoglobin protein. And that is the concept of cooperativity, or cooperative binding of oxygen. So the oxygen molecules, if you like, are cooperating with regards to loading onto the hemoglobin. When one binds, it makes it easier for others to then get, so they're cooperating. Okay, right, so that's one very important concept with regards to the binding of oxygen by hemoglobin. The next really important concept is the concept of oxygen saturation and oxygen saturation curves. So let me firstly explain what is actually meant by oxygen saturation then. So, in a certain volume of venous blood, you're going to have a large amount of red blood cells, and those red blood cells are going to have loads of hemoglobin proteins inside of them, okay? Those hemoglobin proteins are all going to have sites where they can bind to oxygen. So overall, there are a certain number of oxygen binding sites in a volume of blood. The fraction or the percentage of those oxygen binding sites that are actually occupied by oxygen is what we mean by the oxygen saturation of the blood, or just the saturation of the blood. So this is what is meant by this very important concept of oxygen saturation, the percentage of the hemoglobin oxygen binding sites that are occupied within that blood. And of course, what we're interested in finding out is what is the oxygen saturation of venous blood. Well, I'll start by just telling you it, and then I'll tell you about oxygen saturation curves. So you might think that the oxygen saturation might just be 0. However, it's not. The oxygen saturation in venous blood is very high. It's, in fact, equal to 75%. So this is the notation that we use for oxygen saturation. So we have here S for saturation, subscripted V here for venous, okay, because we're talking about the venous blood, and then we've put O2 for oxygen. And this is actually equal to 75%. So what I am telling you is that in venous blood, 75% of the oxygen binding sites on the hemoglobin proteins in the red blood cells are going to be occupied by oxygen. It's going to be much higher in arterial blood. It's going to be 97.5% in arterial blood, but it certainly is not, it's not 0 in venous blood. There is a lot of oxygen present within venous blood. Okay, so now we've talked about oxygen saturation. Let's now go on to the topic of oxygen saturation curves. So what is meant by an oxygen saturation curve? So I said that the amount of oxygen that is bound to hemoglobin proteins is going to be in equilibrium with the amount of oxygen that is dissolved within the blood. And that's true. The amount of oxygen that you have bound to the hemoglobin proteins is going to be utterly dependent upon how much oxygen you have dissolved in the blood. The more oxygen you have dissolved in the blood, the more oxygen you're going to have bound to hemoglobin. That makes sense. Okay, the oxygen molecules can only bind to the hemoglobin protein, and I'll just go up here. The oxygen molecules can only bind to the hemoglobin binding sites if the oxygen is actually already dissolved within the blood. So how many of these binding sites are going to be occupied is going to utterly depend upon what the concentration of oxygen that you've got dissolved in the blood is. And that's what oxygen saturation curves capture. They are a curve which shows for a certain concentration of oxygen dissolved in the blood, what is going to be the oxygen saturation. Okay, so it's a very simple graph. So I will draw a pair of axes then, like so. Okay, so the x-axis then is going to show the concentration of oxygen that is dissolved within the blood. Now, because concentration is complicated and because we much prefer using partial pressure, generally on these curves people use partial pressure. So instead I'm going to put the partial pressure of oxygen. Now note this is not necessarily referring to venous blood or arterial blood, so I'm not putting PA or PvO2, I'm just putting PO2, the partial pressure of oxygen in your blood. So we'll have 0 here, we'll have 20 here, we'll have 40 here. Again, that captures the value for venous blood. Remember, the partial pressure of oxygen in venous blood is 33 millimeters of mercury or thereabouts. And we'll see later on that the partial pressure of oxygen in arterial blood is 100. So we just need to go up really to 100 there. But you might see it going further. And since I've got a bit of space, I'll just go up to 120 there. Okay, so hopefully this makes sense to you. Hopefully this axis now completely makes sense. This is the partial pressure of oxygen in your blood. So this is referring effectively to what concentration of oxygen do you have dissolved in the blood. This is of course really talking about the partial pressure of oxygen in a gas that will create that concentration of oxygen dissolved in the blood. Okay, then on the y-axis what we're going to have is the saturation of oxygen, and again because I'm not talking about specifically venous blood or arterial blood, I'm just putting SO2 rather than having the subscript as I did up here. Do not confuse this with solubility of oxygen. Remember, for solubility of oxygen I had it subscripted. Here I've got the SO2. O is written the same size as S, and those of course confuse this for sulfur dioxide or something like that. Okay, so this is just percentages then, so we'll have 100% at the top, we'll have 50% in the middle, and then 0% right at the bottom. Okay, so what does this curve then look like? Well, the experimental fact is that this looks like a sigmoid curve. So, if you start off then with a partial pressure of oxygen of 0 millimeters of mercury, so we start off right down here, and I should have put some units on here, so this is in millimeters of mercury, right? So if you start off with a partial pressure of 0 millimeters of mercury, then of course you've got no oxygen dissolved there, so of course the saturation of the hemoglobin binding sites is going to be 0. Now, by 33, which is about here, we know it must get up to around 75. That's the value that I've given you, and I've missed. That's not above 33. So it must get up to around 75, so I need to somehow get through there. And this is what the curve overall just about looks like. So it goes like this, and then we'll sort of converge on 100%, so it will gradually sort of get closer and closer and closer to 100% as you get higher and higher and higher. So this is the classic sort of sigmoid curve. It's sort of like an S-shaped. So this is referred to as a sigmoid curve, where it has this steep portion in the middle here, and then it sort of converges on a plateau up here. Okay, so what this is showing then is as the partial pressure of oxygen in the blood gets higher, the saturation of the hemoglobin binding sites gets higher. That's very common sense. Now, there is a complication here that I need to now tell you about, which is that the oxygen saturation curve for venous blood is actually slightly different than for arterial blood, and the reason is that how much oxygen is going to bind to hemoglobin proteins for a certain concentration of dissolved oxygen is dependent upon what the pH of the blood is. And this is the reason I wanted to discuss carbon dioxide transport in the bloodstream before I discussed oxygen transport in the bloodstream. So remember back to carbon dioxide then. Okay, where is what I need? So Carbon dioxide, the dissolved concentration of carbon dioxide is higher within the venous system, over here. And that therefore means that the proton concentration is also higher in the venous system, so the pH is lower, it's more acidic than arterial blood. So there's the basic reminder, pH is lower in the venous system than it is in the arterial system. And I've just told you that oxygen saturation for a certain dissolved concentration of oxygen depends on the pH. And the reason it depends on the pH is because hemoglobin is fundamentally made out of lots of amino acids. I mean, the globin monomers are all polypeptides. They have a huge number of amino acids. A lot of the amino acids have side groups that can absorb protons, okay? So they can bind protons. So for instance, if you've got basic side groups, so examples being lysine amino acids, they can accept protons, and the accepting of protons causes slight conformational changes in the hemoglobin protein, which then leads to reduced affinity of the prosthetic heme groups for binding to oxygen. Okay, so when the pH is lower, which means that the proton concentration is higher, that will mean that more protons are bound to the amino acids of the hemoglobin protein. That will reduce the affinity of these binding sites for oxygen, and therefore it's going to affect the oxygen saturation curve. For a certain dissolved concentration of oxygen, you are going to have a lower oxygen saturation for the hemoglobin. So let's just go down and do that. So what will happen then is for arterial blood, let's say this one is for venous blood here, or rather, well, yes, this one is for venous blood. So for venous blood, because the pH is lower, because of the more carbon dioxide, it's going to have slightly lower affinity for, the hemoglobin is going to have lower affinity for binding oxygen. So now let's draw the one for arterial blood, and I'll do this in a different colour. It's going to be slightly to the left because the arterial blood is going to have higher affinity for binding to the oxygen because it's got the higher pH. So here, and it's only slightly to the left, like so. This now might be the oxygen saturation curve for arterial blood here. Slightly shifted to the left, and remember this means that for a certain concentration of oxygen dissolved within the blood, you are going to have a higher saturation of oxygen in the hemoglobin binding sites. Okay, and this shift of the oxygen saturation curve to the left in this way, when the pH is higher in the arterial blood, the acid concentration is lower. This is known as the Bohr shift. So this difference in these two oxygen saturation curves between venous and arterial blood, this is called the Bohr shift, and it is significant, but I'll talk about how it all works to help with the transportation of oxygen around the bloodstream in just a moment. But for now, know that depending on pH, you will have different oxygen saturation curves, and this is significant because arterial blood does have a higher pH than venous blood because it's less acidic due to the lower concentration of carbon dioxide there. Okay, right. So just before we now go on to actually how oxygen is going to be transported around the bloodstream, let's just comment about how much oxygen then is actually present within venous blood attached to hemoglobin. So we've seen that the oxygen saturation of the venous blood is 75%. So if I was to take all of the oxygen molecules off those hemoglobin proteins in the venous blood, how much oxygen would I actually end up with for, let's say, a litre of blood? So the statistic is that actually in venous blood there is 152 millilitres of oxygen per litre of blood attached to the hemoglobin. So if you take a litre of blood, venous blood of course, and you were to remove all of the oxygen molecules that are bound to hemoglobin proteins in that venous blood, you would get 152 millilitres of oxygen. Now, acknowledge how much bigger that number is than the amount of oxygen that is actually dissolved in the blood. So if we go back up to oxygen dissolved in the blood, it's up here, 1 millilitre per litre. So you'd get 1 millilitre of oxygen out of a litre of venous blood from the stuff that's actually just dissolved in the blood. If you were to take all of the stuff that's attached to the hemoglobin, you'd get 152. So add those two together, overall there's 153 milliliters of oxygen in a litre of venous blood. Okay, right. So now let's actually go on to the transportation of oxygen around the bloodstream. So back to our picture of the cardiovascular system here. So we have now studied oxygen in the venous system, and we've seen that there is oxygen dissolved in the venous blood and there is oxygen bound to hemoglobin. So what will now happen is this blood in the venous system will go up to the pulmonary capillaries. And what will happen in the pulmonary capillaries? Well, of course, the basic stuff tells us that oxygen is going to enter from the alveoli into the pulmonary capillaries, but why is what we need to understand. So we need to know what is the partial pressure of oxygen in the alveoli. So the partial pressure in the alveoli of O2 is actually around 100 millimeters, oh dear, of mercury. Brilliant, just about fitted it in. Okay, so in the alveoli, we have a partial pressure of oxygen of 100 millimeters of mercury. Remember that the partial pressure of oxygen in the venous system is around 33 millimeters of mercury, so this is higher. So that means that you're going to get oxygen coming in from the alveolar spaces and dissolving in the blood. So what is the concentration of oxygen that's dissolved in the blood going to go up to? So we need to go back to Henry's law. So I'll just find this. So here is our Henry's law for oxygen here, and we know that the solubility of oxygen in blood at 37 degrees Celsius is 0.03 milliliters per litre. Times this by 100 millimeters of mercury, we're going to get 3 milliliters per litre. So it has actually tripled from venous blood. Okay, so let me just write that down. So the concentration of oxygen then in the arterial blood is going to be 3 milliliters per litre. And of course, the other way that we could communicate that is by saying that the partial pressure of oxygen in arterial blood, so back to little a there, is 100 millimeters of mercury. And of course, that's the same as the partial pressure of oxygen in the alveoli. Okay, right. So the concentration of oxygen that's dissolved within the arterial blood is going to be higher than venous blood because of the high oxygen partial pressure within the alveoli air spaces, which is maintained there by ventilation, of course. So next question, if we've raised our dissolved concentration of oxygen up by a factor of 3, how much are we going to raise the amount of oxygen that's carried on our hemoglobin? So the oxygen saturation is going to now go up because of two factors actually. Not only because we have increased the partial pressure of oxygen of the blood, okay, so one reason is that we're going along here, we're going up now to 100, okay, so that's one reason that the saturation is huge. Oh, whoops, that's horrible. Let me just reverse that. Try and actually draw a straight line. So one reason is that we've gone up to now having a partial pressure of oxygen of 100 millimeters of mercury. So of course we've moved up from 33 here. Okay, the other reason is of course that carbon dioxide is coming off. It's coming out of solution at the pulmonary capillaries. Carbon dioxide is going out of the blood, and therefore the pH is going to go up. It's going to become less acidic, and therefore hemoglobin is going to be deprotonated. The affinity is going to go back up, so we're going to have the Bohr shift occurring now. So we'll go to this red curve, and that's another reason that the saturation of oxygen is going to increase because of the increase in pH, the decrease in the proton concentration. Okay, so what then is the actual arterial oxygen saturation going to be then? So the saturation of the arterial blood of oxygen is actually equal to 97.5% usually. And that actually corresponds to carrying around 197 milliliters per litre of oxygen. So if I was to take a litre of arterial blood and take all of the oxygen that's bound to hemoglobin proteins in that litre of arterial blood, I would get 197 milliliters rather than 152 milliliters. So add that to the 3 milliliters per litre that is dissolved within the arterial blood, and you get that there's around 200 milliliters of oxygen per liter of arterial blood. So oxygen is indeed higher in the arterial blood than it is in the venous blood. Remember, it was 153 milliliters per liter in the venous blood. Okay, right, so completing up the story, so going through the pulmonary capillaries will turn the venous blood into arterial blood, and we've now seen that both the oxygen concentration that's dissolved has gone up, and also the actual oxygen that's bound to hemoglobin has gone up. So the partial pressure, arterial O2, is higher, and the saturation, arterial O2, is higher. What's now going to happen is, of course, we're going to go through the peripheral capillaries, and here oxygen is going to leave. Now, why is oxygen going to leave? Well, the reason now is that the concentration of oxygen dissolved in the tissue fluid is much lower than the concentration of oxygen dissolved in the arterial blood. So hopefully you can figure out what the oxygen concentration in the tissue fluid is going to be, and I'll get back my pink pen that I use for tissue fluid. So the concentration of oxygen in the tissue fluid is going to be, of course, the same as the concentration of oxygen in the venous blood, because that's the reason that you got that oxygen concentration in the venous blood. So it's going to be 1 milliliter per liter, and equivalently we could use the partial pressure of oxygen in the tissue fluid is around 33 millimeters of mercury. So what, of course, will happen is oxygen will move from the peripheral capillary into the tissue fluid, and then, of course, into the actual cells, hopefully. And that will mean that the oxygen concentration that's dissolved in the blood will go back down to the venous concentration. When you do that, of course, we're moving the other way now on our oxygen saturation curve. So back to the oxygen saturation curve down here, we're moving back down now to the depths of 33 millimeters of mercury being our dissolved oxygen concentration. And of course, also at the same time, CO2 is being dumped into the blood in the peripheral capillary, so the pH is going back down, it's becoming more acidic, so we're going to go back to being this blue curve. So that's the other reason that the oxygen saturation will go down, and it will go down now to 75%, so you've had a lot of a lot of oxygen taken out of the blood. And then we're back at the venous blood. So there is the story of how oxygen is transported around the bloodstream. So to end the video, there's one little calculation that I'd like to do. So what I would like to calculate is how much oxygen is actually going to be delivered to peripheral tissues in a minute. And this has a name, and of course this is at rest. This has the name VO2. So this means volume of oxygen consumed in a minute, or we can just call it oxygen consumption. So how much oxygen are we going to deliver to our peripheral tissues in a minute? Well, let's think about this. Remember our cardiac output, the amount of blood pumped around the body in a minute, CO, it's usually around 5 litres. So 5 litres a minute of blood is pumped around the body. We know the difference in the amount of oxygen carried in arterial versus venous blood, and we know how much blood is going around the body in a minute. Therefore, we can work out how much oxygen is being delivered to the tissues in the body per minute. So how much oxygen was in a litre then of arterial blood? Remember, it wasn't 197 quite. That was the amount of oxygen carried on hemoglobin in arterial blood. We have to add that to the amount dissolved in the blood, which was 3 milliliters per litre. So, and I'll get another colour to make it a little bit more interesting. We will have 200 then milliliters of oxygen per litre in arterial blood. Now let's think about how much we have in a litre of venous blood. Well, 152 milliliters, again, that was the amount on the hemoglobin protein. So we have to add on to that the amount that's just dissolved in the venous blood, and that's 1 milliliter per litre. So this becomes 153 then. So 153 milliliters per litre is the amount of oxygen in a litre of venous blood. So subtract those off, and that's the, subtract this one from this one, and that will give us the amount of oxygen per litre of blood that's been delivered to the peripheral tissues. That's per litre. We are delivering 5 litres of blood to the peripheral tissues in a minute, so we just need to multiply that difference by 5. So take this and just multiply it by 5 litres, and you can see that all of the units are going to add up here. So taking 200 and subtracting 153, you'll get 47 millilitres per litre. And then we times that by 5 litres, and this now gives us the volume of oxygen that we're delivering to the tissues in a minute. So doing this, 5 times 40 is 200, 5 times 7 is 35, so we get 235 millilitres in a minute. And this is the normal oxygen consumption, the normal amount of oxygen that's going to be delivered to the peripheral tissues in the human body in a minute. Okay, and there we conclude our discussion of the transportation of oxygen and carbon dioxide around the bloodstream. Thank you for watching this video. I hope you've learned something.